Well, good evening, everyone, and welcome to the annual Robinson Lecture. As you know, this is an annual event where we welcome a speaker who is a former member of the college, a member of the college, really, because you're a member for life, and one who has gone on to some distinction in their career, to come and share their experience and tell us about their current work. So, our speaker today is Professor Wendy Barclay, who is currently head of the Department of Infectious Diseases at Imperial College, London. Professor Barclay studied natural sciences here at Robinson before doing a PhD from the University of Reading while working at the Common Cold Unit. She's worked for many years on influenza viruses, their mechanisms of transmission and replication, and was, in the years before the COVID pandemic, she was a persistent voice calling for pandemic preparedness. Of course, the disruptive pandemic that hit us 4 years ago was not a flu pandemic, but the expertise of virologists such as Professor Barclay was called upon both to advise official bodies, she served on NERVTAG and SAGE, and also to inform the wider public. She became an expert on SARS-CoV-2 as well. She was awarded a CBE in 2022 for services to virology. So, Wendy, it's a pleasure to welcome you back to Robinson, and over to you.
Thank you very much for the kind introduction. It's a real honour to be here. I haven't been back very many times since 1985, but the college is remarkably similar. It's still got bricks all over the place, and I'm actually staying tonight in the staircase K, and I spent 2 years of my life on L5, so it's not very far away from where I grew up, which is lovely. I'm going to talk this evening about what I'm up to at the moment and how I got there, and I'm purposely being a little bit controversial in the parts of my work that I'm going to tell you about, because I hope that will stimulate lots of interesting questions and some discussion later. I didn't ever intend to be a virologist. I didn't study virology while I was here. My final year, part 2, was pharmacology. I thought that I was going to make lots of money going into the pharmaceutical industry. Had a few interviews on the milk round and utterly failed in all of those. But I had applied, meanwhile, for one PhD position, which was a very peculiar description of an opportunity at a place called the Common Cold Unit, where people volunteered to go and get given on purpose a common cold by sticking liquid-containing virus up their nose. And that was what I did for 3 years and studied them intensely. And I'll talk a little bit about what that's led on to in more recent times. So when I graduated and took that PhD position, I didn't really know what a virus was. It was 1985. The first ever crystal structure of a virus particle was on the front cover of Nature. That was unbelievable. I mean, the timing for me was almost perfect. It was the moment at which people began to really understand how you could pick apart the nuts and bolts of what a virus was and how it worked. These days, these are the sorts of images that my lab can produce, and many labs can produce these, but I think they're amazing, and they really sum up, for me, the fascination of viruses. So this pink thing is a cell from the lab, and about 8 hours prior to this being taken, it was infected with probably one single viral particle, and all of those purple-coloured protrusions you see are the progeny viruses emerging from that cell, hundreds or if not thousands of them from each individual cell. And you know, the capability of this self-replicating entity, which is so small we can't see it without using these electron microscopes, to cause the devastation across the world that we've seen in the past 4 years from COVID, I think is absolutely phenomenal and still fascinates me to this day. You can make one of these things in the lab. That's a controversial thing to say, but you can. And you know, it can produce all this disease and devastation across the world. But as I say, I did not intend to be so excited about viruses. Here I am looking wistfully out at the image from my balcony, just about to graduate. Here's myself and two of my very best friends when we were in college, pretending, I think, to revise for our finals. But that patch of grass was a very well-worn and very nice sunny place on a May afternoon to meet up with your friends and not really do all that much revision. And as I say, at the end of that 1985, having graduated from here, I sort of ended up by chance, really, not by any planning, at this place, the Common Cold Unit, which was also known as Harvard Hospital. The history is that in the Second World War, the Americans and the British became very concerned about the concept of infectious disease and how it might spread in places like air raid shelters where people were being brought together in very close proximity. And everybody decided that much research was needed and that common colds would form a good model for this kind of thing. And in their wisdom, the Americans decided that for some reason that the British couldn't build a place to do this. They thought we should study it on Salisbury Plain, but we couldn't be responsible for building it. So they shipped these prefabricated buildings over from America on boats from Harvard, and then set them up here and sort of let it happen. And so this was run for years by the Medical Research Council. And as I say, volunteers would come down from all over the country and have their holiday in Salisbury being inoculated with a virus. So I'll talk a little bit later about human challenge studies and what we can do with those. But let me sort of begin by saying what I'm going to talk to you about today is really pandemics. We've just lived through a pandemic. They're very familiar now to everybody. But as I say, I'm going to talk a little bit about some of the work we've done, sort of a few snippets of the work, I think, which I probably enjoy talking about most of all because it is slightly controversial and on the edge. So the first thing I'll say is that just because we've recently lived through COVID does not make it any less likely that there will be another pandemic. You know, just because you win the lottery once doesn't mean to say you're not going to win it next week or the other way around. So the questions that I ask are, could we prevent it? And if we can't prevent it, what would we do differently? What can we take from this experience? And the sorts of things that we need to understand is, well, then how do these pandemics arise? Where do they come from? And in that case, by knowing that, can we minimise the opportunity for the virus? And then how do they spread between us? And I will try and persuade you that understanding transmission of viruses between people is probably the key to early control and getting a grip on all of this. So can we prevent spread better in the first wave of a pandemic than we did last time around? I think everyone in this room, and certainly every person on the street, every member of the public now knows that pandemics come from animals. Viruses that infect animals can cross into humans, and that's how we get pandemics. The two viruses that have driven my life in the last few decades are the one on the left, which is influenza. And influenza is not a virus of humans, it's a virus of birds. It resides in many different forms in these birds that you see here, these wild water birds, ducks and geese. You can see that this is a fantastic place for a virus to hang out because there are many, many hosts which it can infect, and they all congregate together in one place on the lakes, particularly if they're migratory birds. So the virus is spread through the water, it infects the gut of the bird and it's shed into the water, and then its cycle is perpetuated. Coronaviruses, on the other hand, on the right-hand side here, this is SARS-CoV-2, often are found in bats as the reservoir. And you can see once again what you have is a population of hosts which are very numerous and very dense, and therefore the virus doesn't have far to go from one host to the next in order to perpetuate its cycle. And, you know, if we didn't get in the way, that these viruses are highly adapted to these animals, they cause very little disease in these animals, and it's a perfect sort of symbiotic relationship. The issue is that sometimes viruses have a conduit from those wild animals towards humans, and usually because what we've done is come along and domesticated animals for our own purposes by farming, chickens, pigs, or perhaps having pets, and viruses which are in animals then can reach us through those intermediate species. Here you can also see on the bottom here the Hunan market, now famous in Wuhan, the place where people can go and buy animals as meat, either as meat or as live animals, which is a popular thing to do in Southeast Asia. And then I'll talk a little bit more about these creatures a bit later, but these are mink. And of course, these are another example of animals we farm, not for their meat, but for other purposes. So influenza is really our paradigm for a pandemic virus. We know that there is a numerous wild animal reservoir. There are many different flavours of the virus, none of which have infected humans before, apart from the 2 or 3 that we know about that are the seasonal flus that were once bird viruses but crossed into humans many decades or even hundreds of years ago. And most of the time, those viruses sit in those birds. They don't cause any problem to us. We don't have close enough contact with a duck for it to really pass its virus to us. But those duck viruses can find their way into chickens or pigs or both. And those infections in domesticated animals with which we as humans have a much larger interface can be then the conduit or the intermediate, the stepping stone for the virus to reach us. And we know that influenza has a documented record of causing pandemics, 1918, 1957, 1968 and 2009, 4 in about 100 years or so. So it wasn't very surprising that nerve tag was formed after 2009. Everybody was sure that in a densely populated world with lots of farming going on, there would be another influenza pandemic. And most of nerve tag's time was spent thinking about, were we well prepared for the inevitable influenza pandemic? And quite rightly so, because at least there are things we had in our armament to think about, like vaccines and antiviral drugs that could be useful in a pandemic, and it would be important to understand if indeed they should be purchased and stockpiled and distributed. The reason that we don't get infected all the time with these avian viruses is at 2 levels. One is that, as I've pointed out, the way the virus goes from bird to bird is really very different from a physical perspective than the way that viruses go from human to human. Viruses pass between humans, at least influenza does, in airborne droplets, airborne transmission. And the change there means that viruses which are well adapted for transmitting through water between birds don't readily then just jump into humans and just take off as pandemics. There are many molecular changes in the virus's behaviour that will result in that. But the other reason is that viruses are what we call obligate intracellular parasites. They're very, very simple. As I've said, they're so small you can just make one in the lab. They carry into a cell that they're going to infect the minimal amount of genetic material. And then inside the cell, they hijack all the cell's machinery and co-opt it for their own purposes. And that's why those hundreds or thousands of little viruses that you saw in the first slide were so successfully coming out just a few hours later, because they had taken over the entire cell machinery and using it for their own end. But what that means in terms of using cell machinery is that cells are different between different species. Human cells are not the same as avian cells. There are different proteins and other factors inside the cells which the virus needs to grab hold of, which will be different. And so if an avian virus infects a human cell, it's really not very good at taking over those other factors it needs inside the human cell, because there's an incompatibility, a mismatch that is as a result of years of co-evolution with its first host, which means now it's not well matched to its new host. And that slows the virus down, and it's the reason why most of the time, even if we did come into contact with an infected bird, a duck or even a chicken, we would not really get infected productively with an avian influenza virus, because the virus is just not capable, unless it mutates, to take over a human cell. Now, I have to show you this because this is the pinnacle of my career. In 2016, I managed my one and only senior author Nature paper, and I'm very proud of it. And what my group did, thanks to a wonderful postdoc who worked in my lab called Jason Long, was discover one of these proteins which sits inside the cells of humans or pigs or birds. And when influenza virus infects those cells, it utterly relies on grabbing hold of this protein and using it for its own purposes. This protein sits in the nucleus of the cell, And this little schematic is basically describing the fact that the bird version of the protein, the avian version in orange, is different than the mammalian version, and that's one of those incompatibilities that I was describing. So we were able to account for what we call the host range barrier, or the species difference between humans and birds, which mean that we don't readily get infected with avian influenza. And we could also show that the virus itself can mutate and switch one amino acid in its polymerase there from a E, a glutamic acid, to a K, a lysine. And that allows it now to use the mammalian homologue of this protein. So this explains that host range barrier. But what it made us realise is that ANP32, this protein, is very important if you want to understand influenza. Now, these days, our colleagues at the University of Oxford have solved the crystal structure, which I think is, again, one of these beautiful images which I never dreamed I'd have anything to do with. But this orange blob in the middle is the host protein, and it's cross-linking 2 units of the viral polymerase. And in doing so, it's sort of completely, the polymerase has hijacked this protein, has sort of sandwiched it in the middle, and is making the protein help it replicate itself inside the cells. If that protein isn't present in a cell, then influenza is completely incapable of replicating inside the cell. It's absolutely essential for the virus to be able to grab hold and sandwich that orange protein there. And we showed that, for example, in cell cultures by taking the protein away and showing that those cells were refractory to infection. You could put as much virus on them as you liked, and they just didn't incubate and replicate the virus. And that made us think, could we use this information in a very different way than we thought of before? So how about if we were able to alter the genetic code in a chicken? And of course, chickens are very susceptible to avian influenza. They can be the conduit for the virus to travel from birds or wild birds through to humans. What if we could make the chickens resilient? So if we could take those chickens and stop them from being able to make any bird flu, we would not only help the chicken farmers, because they wouldn't have to cull their chicken flocks when they got bird flu anymore, but we would also protect humans from that conduit, that pathway towards pandemics. Now, we've been mucking about with chickens for years, using forward genetics, animal breeding. So you may not know, many people in the room will have never seen a chicken like the one in 1950, but that's what we used to try and eat. These days, our chickens are a lot fatter. They have been bred over the years to be very different than the jungle fowl which they started off at. And actually nobody really minds that. I mean, that's what happens. It is an issue for animal welfare, which is important. But nonetheless, this is a very accepted thing. However, making transgenic animals which we might then eat is not currently very accepted by the public. So these little chicks here have been engineered to express the green fluorescent protein from the jellyfish. They express it all over their body, but in fact you can see it best in their beaks and their feet when you shine the light on them. This is old-fashioned transgenesis. So this is what people have been doing in plants and animals for many, many decades now. But it is unacceptable because when you do this sort of thing, you introduce extra pieces of genetic information that could be immunogenic, could be harmful to people. What we have today is very different because since Jennifer Doudna and Emmanuel Charpentier invented CRISPR, gene editing, we can now precisely alter the genetics of animals and plants in a way which means there's nothing extra there. You know exactly what you've done, and you have the capability to engineer precise changes without any others coming along. And in fact, this has already been used, for example, to generate these pigs, which are resistant to another virus called PRRS, which is devastating in China. And not only that, but of course the UK, following Brexit, is allowed to make its own rules and regulations about these sorts of things now. And in fact, the Genetic Technology Precision Breeding Act was passed by Parliament last year, which means that it is now legal, if you like, to precisely engineer plants and animals for the purposes of food. So with all that in mind, we decided that we would give our gene-edited chickens a go. we reached out to the Roslin Institute in Scotland, which is the world's best place really for making gene-edited animals. They were the first people to produce, for example, Dolly the sheep, and they know all about farmed animals and what you can do with them. And they generated for us gene-edited chickens altered in our ANP32 host factor, the one which is essential for influenza. And there are 2 chickens in the slide there, and I challenge you to be able to tell me which one of those is wild type and which one of those is gene-edited. So I can reveal that the one wearing the little anklet, the blue anklet, is gene-edited, but you couldn't tell, the farmer couldn't tell, the public couldn't tell. These animals grow as fast, they lay as many eggs, they make the same responses to vaccines as wild-type birds. There's nothing in them different other than just 2 nucleotides in their entire genome which make 2 amino acid changes and make their ANP32 less good or almost unable to be used by the influenza virus when it infects them. And this is the paper we published on that, but I can sum it up to say it was all looking quite good. So this is the experiment we did. We took some wild-type chickens, illustrated there in brown. We infected them with 1 million infectious units of virus, which is an awful lot. And then we put in the same incubator either 8 other wild-type chickens or 4 gene-edited chickens in green. And what you can see is that all of the wild-type chickens got infected. There was lots of virus being transmitted about in that incubator, but none of the gene-edited chickens became virus positive. So in this scenario, if we had gene-edited chickens in the farm and a duck came along and tried to transmit its virus to our farm chickens, they would be resistant to infection. We wouldn't incubate infection in the poultry farms. We wouldn't have to cull all the millions of birds that we end up having to cull. However, we pursued the experiment a little bit more, like scientists do, just probing, pushing the limits, the boundaries. And what we found is that if we took that same infectious dose, that 1 million infectious units, and put it directly into the gene-edited chickens, we could break through the infection. We could actually make that virus infect to low levels, but we could pick up virus in 5 of 10. Still none of the 8 gene-edited chickens in the same incubator got infected, but one of the 4 wild types did. And this told us, back to the drawing board, I'm afraid. This is not acceptable. So this is a controversial thing to be trying. I'm not trying to persuade you that this is the answer. But what I can tell you is that there are scientists all around the world now considering whether or not gene editing of animals and plants is the way that we want to go in order to feed the 8 billion people on this planet. And what is important is that we do robust science and push the boundaries and responsibly communicate that information to other scientists, to the public and to regulators, so we can have a healthy and informed discussion about whether or not this is what we want to do. So we've published this work. We've said, look, a single edit like we introduced here is probably not good enough. What we need to do is to set up a protocol which we all agree to, which is how will we assess this going forwards and how will we decide when or if gene editing animals is going to be acceptable. So that's my first little controversial story to explain to you. Meanwhile, of course, there are other animals and other routes by which avian influenza virus could reach humans. And one which has really come to the fore in just the last few years is through a different sort of farming, which is mink farming. So we don't do any mink farming here in the UK. It is illegal, but we do, of course, import mink coats and sell them in our stores. That's not illegal. And there are many places in Europe which still farm mink. So Spain, Denmark, Poland. Just over a year ago now, there was a bird flu outbreak in a Spanish mink farm, which was really quite worrying. So there is, I will talk about in a little while, a strain of bird flu going around at the moment with a snappy name of H5N1 clade 2344B. It is currently doing what we call panzootic, so it's all over the world and it's killing many wild animals. You will have heard of it on the news. It's in the penguins now in Patagonia and elephant seals as well. Many wild bird species have suffered huge population losses as a result of this virus in the wild, but it has also incurred into poultry and now mink farms.
what the worry is that mink are a known mammal which can adapt or drive evolution of the virus in a way which makes it more likely to infect humans. And so the worry is that when an avian flu virus gets into a mink farm, this is a dangerous situation in which a new pandemic could be incubating, and we probably should be thinking about minimising this. Many of these mink farms have very poor biosecurity, and so there is an obvious route for wild birds to access these animals and introduce their viruses to them. We know that mink can also not only incubate human viruses, but they can pass them back to humans. So back in November 2020, actually the very first of the SARS-CoV-2 variants was picked up in a Danish mink farm, where the virus of the time, even before Alpha, this was an early virus, had got into the mink that were being farmed there, and then people in that village had then caught the virus back from the mink and taken it home and spread a sort of variant, a mink variant through the village. So there is a two-way exchange of viruses between human and mink, and it is a dangerous situation in terms of pandemic propagation. And so that really prompted myself and one of my other great postdocs, Tom Peacock, to write this opinion article just last year, which was published in the Proceedings of the National Academy of Science of the USA, just pointing out that mink farming poses a risk for pandemic propagation or pandemic emergence. And indeed, this was taken up and was discussed quite recently in Parliament. Okay, so what I've tried to say there is that we could be thinking about ways to minimise human exposure to dangerous viruses like influenza, because that's the obvious route by which a virus would reach humans and become pandemic. Influenza, exposure to avian influenza doesn't always lead to pandemics. Many people, hundreds of people in the last decade or so, have been exposed to avian influenza. They've got very seriously ill, so there's a famous series of viruses in China and Asia in the early 2000s, which cause what we call these zoonotic infections, where people get very ill. But that's still not a pandemic. So a single incident of a human being infected with an animal virus is a zoonosis. Further mutations are required in the virus for then to become airborne, transmissible, and cause a pandemic. And then, of course, don't forget that those pandemics grow up, if you like, or evolve to be seasonal influenzas. So if we think about what these respiratory viruses need to do, they come out of animals, they look new, they have what we call novel antigenicity. They replicate inside human airway cells, but the key that protects us really, the big barrier for all of those viruses to then become proper pandemics is that they would have to mutate and change the way they interact with their environment if they're going to transmit efficiently between people through the air. And something I'll mention in a minute is about whether or not that transmission occurs with, prior, or without symptoms. So as I was saying, in the early 2000s, there was a lot of worry amongst the scientific community and the public about whether or not bird flu was being hatched as a next pandemic in parts of Asia. So H5N1, H7 and 9, these were strains of avian influenza infecting hundreds of people, making them severely ill, and a lot of alarm, but thankfully we didn't see a pandemic arising from those viruses. It's rather similar today. We have this virus I've mentioned, H5N1, clade 2344b. It's panzootic. It's infecting seabirds. It's infecting mammals that scavenge off the dead seabirds. Just this is last week's Telegraph, I think. It's now infecting penguin chicks and elephant seals in South America. Thankfully, only a few humans have been infected by this particular virus so far, and there's been no onwards transmission. But that is all that is separating us from a devastating pandemic which originates from bird flu. So really, the most important thing for us to understand in terms of pandemic preparedness is transmission, the process of airborne transmission, viruses passing from me to you in these airborne droplets that I breathe out all the time. If I cough and sneeze, there are more of them, but I'm breathing them out, and often I can be silently infected, breathing them out and passing them on to other people. So transmission is this droplets which carry viruses across. Of course, they may fall prematurely onto fomites and then be picked up through contaminated surfaces. They may evaporate water and become aerosols. And the reason that their survival and their ability to propagate their infection is really all about how the virus looks to the outside world, what's on the outside of the virus. So pandemics emerge when animal viruses change in a way that means they can transmit between humans. That knowledge and understanding which mutations then can do that and which viruses can accept those mutations and become pandemic is key to being able to decide which of all of these different flavours of influenza virus that are in birds in the wild and which of all the different types of coronavirus which are in bats and other animals in the wild, which of those should we really prioritise? Because with our current technology, we make vaccines which are specific to individual viruses. And we can't make pre-pandemic vaccines for them all. We need to understand which ones to put at the top of our risk list. So a few years ago, some scientists, not us, but two groups of scientists, one in the United States, one in the Netherlands, performed some experiments to try to ask whether those H5N1 bird flu viruses, which were around in the early 2000s, really did or did not have the potential to become airborne transmissible. And they performed a series of experiments where they combined their best knowledge and engineered viruses containing mutations which they knew would adapt the virus somewhat for airborne transmissibility. And then they passaged that virus in what we would call a forward genetic experiment through ferrets, because ferrets are a very good model for human respiratory viruses. And they ended up in the laboratory under very high containment level, creating a virus which had never existed in nature before, but had the potential to become airborne transmissible between ferrets. And this is now termed gain-of-function research. They created a virus which didn't exist in nature and learned a lot about what the mutations are that are required to transform an animal virus into one with human pandemic potential. But this controversy spurred a research moratorium, certainly in the United States and around the world in these areas, and is today highly controversial and probably, if I'm honest, gave rise, or at least fueled, the notion of the origins of the virus which did actually emerge as the next pandemic in 2019, SARS-CoV-2. So SARS-CoV-2 emerged as a novel beta coronavirus in the Wuhan area, as we know, and it emerged already with efficient transmission between humans. Let's just look at the timeline. So SARS-CoV-1 had been around in 2003. It took a long time for the WHO to be informed about that virus, but thankfully, due to what we call non-pharmaceutical interventions, the first SARS virus was controlled. We were able to identify people who were infected with the virus because they all became seriously ill, and they didn't transmit their virus to anyone else until they were seriously ill, which meant that you could find the patient, you could put the patient in isolation and prevent them then from passing their virus. Unfortunately, of course, some medics were exposed, and there were some very tragic incidents of healthcare workers becoming infected and either severely ill or dying. But SARS-CoV-1 was controlled. SARS-CoV-2, China informed WHO much earlier than the previous time, but by that time, we'd already lost the battle. This virus is so highly contagious through the air between people that, as we know, we went on and had this explosive outbreak. And really, that's because many people infected with SARS-CoV-2 were not fatally infected or severely infected. They were mildly infected or even asymptomatic, but they were still contagious to others. So we couldn't see easily who was infected at that time. Of course, we didn't have diagnostic tests to know that either. We didn't have lateral flow tests available. And people were walking around in our communities spreading their virus to other people without us knowing it. So what we know is that SARS-CoV-2 is transmitted much more efficiently than SARS-CoV-1. Why is that? Well, there's one obvious molecular difference that scientists could see straight away between SARS-CoV-1 and SARS-CoV-2, and it is known as the furin cleavage site. So the spike protein, which is the protein which is on the outside of the virus, is the way the virus latches onto cells and enters them. And spike has to be activated to do that by cleavage, cutting at a couple of places. And SARS-CoV-2 has this insert in it, which gives it the ability to be more easily cleaved. Now, this cleavage site leads to better replication in airway cells and its ability to infect people much more efficiently and therefore transmit. And I really think it's all summed up by this headline from El País magazine: "These 12 letters changed our world." These 12 nucleotides encode the 4 amino acids which sit and are the furin cleavage site. Human airway cells are the primary target for all of these viruses, and we use them a lot. These viruses come in, they chop through the mucus, they attach to those airway cells and infect them. Using those cells as a model, what my lab was able to do fairly early in the pandemic was to take a pair of viruses, the wild-type SARS-CoV-2, which was transmitting between people very readily, this orange one, and then a lab-adapted version, which just spontaneously arose when we grew the first virus in cells. So that's the blue one. And the blue one had just got rid of that furin cleavage site. It didn't need it to go from cell to cell in the lab, but it needs it to go from human to human. And we could show that by putting the 2 viruses into these special human airway cultures, which mimic absolutely what your nose and throat look like. And we did a competition between the orange virus and the blue virus. And you can see that over just a matter of days, the orange virus completely won the competition. What that says is that the orange virus is much fitter in the human airway than the one that doesn't have the furin cleavage site. So absolutely, the furin cleavage site is what makes the difference for SARS-CoV-2 to be that very efficient transmitted human virus. And we were also able to perform experiments in our model animals showing that that made the difference between transmission. Now, a lot of speculation has gone on largely on social media that furin cleavage site was purposefully engineered into the virus by scientists in the Wuhan lab. I'm here to tell you that as a virologist of this many years, I don't uphold that theory, and I'll explain to you why in this picture, if I may. If I were one of those Chinese scientists in that lab trying to put a furin cleavage site in there, I wouldn't use the one they used, because the one they used is really not very good. The one they should have used is this one here, which we used in the paper that we studied. We didn't make this virus, but you can make artificial versions of the spike protein. And this one is taken from a flu virus. This is the H5 cleavage site. So influenza viruses have furin cleavage sites. It's a well-known trick that all viruses that evolve naturally tend to get these sites. And you can see here the sequence is full of these arginines and lysines. These are the single amino acid letter codes for these very basic amino acids, which is what really makes a furin cleavage site. The one that naturally evolved in SARS-CoV-2 has 2 arginines and it has an alanine and then another arginine. So I think any scientist would hang their head in shame if they'd been asked to insert a furin cleavage site into this virus and put the one that actually was found in it. Not only that, but nature itself has shown us that since our SARS-CoV-2 virus emerged, we have seen a whole series of further evolved viruses, which nobody is suggesting were created in any labs, where this furin cleavage site has gradually acquired more and more mutations as it circulates in humans, becoming better and better at transmitting. So evolution goes on here naturally all the time, using selective pressure to select for the viruses which are the most transmissible. And we don't need any artificial theory of creation to account for how this virus arose. Okay, so SARS-CoV-2 pandemic came. We used largely non-pharmaceutical interventions to try and stop its transmission in the early waves of the pandemic, as we know from the current inquiry, at huge economic cost. What do we wish we knew sooner? Next time around, what sorts of experiments and protocols could we set up? Going right back to the beginning of my early career, one thing that is very informative is to do intense studies, kinetic studies, in people. It's very difficult, though, in natural situations to find people at the moment that they become infected, because by the time they turn up at their doctors or they turn up at the testing centre, they've all been infected by 3 or 4 days, and you've missed a lot of important information. One way you can get around that is by deliberately infecting volunteers, just like we used to do at the Common Cold Unit. But back in the days of the Common Cold Unit, we were infecting them with a common cold, a rhinovirus, a virus which nobody dies of. What we thought about doing in the early days of the SARS-CoV-2 pandemic was to infect people with COVID, with the SARS-CoV-2 virus. And there was a lot of ethical discussion, many backwards and forwards. In fact, this was highly supported at the time by the government and the vaccine task force at the time, because in mid-2020, we knew that many hundreds of different vaccines were being developed all around the world. And there was no way that there was going to be time to run enough clinical trials to test them all at the capacity required for a clinical trial. But the idea that one could triage those vaccines in some sort of experimental medicine system, like a human challenge study, and then take the very best ones forward to clinical trials was a very popular idea. So funding was put forward to set up the first and still the only human challenge study with SARS-CoV-2. Young, healthy adults were known at the time to largely not be getting what we call full-blown COVID. They were getting mild symptoms or even no symptoms at all. By that time we had good testing, we could see that most healthy people were not getting sick. And so we put out a notice. Thousands of people came forward as volunteers, and from all of those thousands of people, 36 were selected based on their fitness, their health, and they were deliberately inoculated. You can see one of them receiving their virus there into their nose with 10 infectious doses of SARS-CoV-2. And many, many interesting studies have happened looking at the immune system of those people and what happened next. But what we were interested really in my lab was when they get infected, when do they become transmissible to other people? At what point in their infection do they start putting virus out into the air with the potential to infect the next person? And so here's an image of some of the data, very detailed data. This is a time course here of all the days since they've been infected. So day 0, they get infected, and we monitor them for 14 days. We measure virus in their throat. You see that comes up first in the blue. We then measure virus in their nose. We measure virus in masks that they wear and in the air that we sample from the air and on their hands and on their environments. This volunteer here, participant 7, is one of the 18 who got infected. Very interestingly, the 18 didn't, and they had no vaccine and no prior exposure. So how do we know what was protecting them? Because we physically stuck the virus up their nose and they still didn't get infected. That's another story. Participant 7, you can see here, didn't really put any virus out into their breath or the environment until they became positive in their nose. So one thing I can tell you is when you see people wearing face masks slung around their chin or just over their mouth, that's really not helping you, because what you want them to do is to block their nose off, because the virus which is coming from their nose if it's going to come and infect you. We can also see that this person had a symptom score, and on the days when they were putting out the most infectious virus with these solid black bars into their mask, they experienced absolutely no symptoms whatsoever. So this person would have been walking around in the supermarket not knowing they were infected, breathing out infectious virus onto other people. And this is the problem for SARS-CoV-2 and for controlling an outbreak like this. What we absolutely need is a better diagnostic kit, which is easy to use at home, by which people can identify when they are contagious to others. And at the start of COVID, I can tell you that most professionals would have told you that the general public are not capable of using such diagnostic kits, lateral flow tests. But most school children can do it just fine. And what we show here is that if everybody were to take a lateral flow test every day and not go out on the days they're positive, there would be no transmission. Okay, the dotted line here is the day that they become lateral flow positive, and all the green is when we infect them, we detect them being contagious. So if they just stayed home the day that they become lateral flow positive until they become lateral flow negative, then we would not need lockdowns. We could have self-informed isolations. The other thing we found, which was very interesting, was that just 2 people out of the 18 that were infected put out 90% of the virus. Now, this is a bit controversial, but if only we could tell who those are, then those could get locked down and the rest of us could carry on down the pub. Unfortunately, we can't at the moment find any parameter which identifies those people, and probably that's a little bit controversial as well. But I think having ways to understand the basis of this heterogeneity and what sorts of differences. What we can say is that when we did original breath tests, one of those 2 people scored the highest in their breath tests. They were big breathers. So singing, exercising, things that help you breathe big, of course, are what they're, but some people just naturally breathe big, and that can make you a bit more contagious than everybody else. What's happened since? Of course, wave after wave of SARS-CoV-2 variants. So what I currently spend my time doing is leading a consortium of virologists around the UK trying to work out, should we be worried about any of the new variants, Omicron, BA1, BA2, EG51, JN1.1, they keep coming. Thankfully, the vaccine seems to be holding in terms of protecting against severe disease mortality, but we're not resting just yet. These viruses evolve all sorts of different ways, and we have to keep looking at this for a little while longer. And I'll finish with a sort of final remark, which is that in doing all of this, of course, and the way that the public currently feel, with social media perpetuating the concept that scientists are to blame for everything here, means that a lot of the work that we do to try and pull apart the nuts and bolts of the virus and understand which of those variants we should or shouldn't worry about gets us featured in places like the Daily Mail. I don't mind so much, but these are my two wonderful postdocs who work night and day trying to do their very best, and here they are being accused of very bad things. Of course, everything we do is highly regulated by the Health and Safety Executive, done in high containment laboratories with oversight at many levels. But nonetheless, I think it's important that we start communicating that oversight and the way that it's done better than we currently do. So I hope I persuaded you that the most important thing to understand in a pandemic is transmission. We need to keep an eye out for the animals that we have a lot of interaction with. Don't underestimate the public in terms of what they will do. I mean, people can do a self-swab. And just imagine in the next pandemic, if everybody had one of those, or many of those in their bathroom cabinet, they had a reporting app we could monitor around the country, they had a stockpile of antivirals to suppress their virus and help them when they needed to, and even block household transmission, individuals could make informed decisions. And I think this is the way forwards, that the public might accept some level of regulation in the next pandemic. And communication, as always, is just going to be key to win and maintain public trust in science, particularly virology, as we go forwards. So thank you so much for having me here. I'd be very happy to talk about some of the controversial things that I've mentioned. I've got a wonderful team of young people that work with me. I've got wonderful collaborators, and I've been funded over the years by all of these funding bodies without whom I couldn't do anything in the lab. Thank you so much.
Okay. Well, thank you very much, Wendy, for that talk. So we have time for some questions. If you'll raise your hand and wait for the microphone to come to you, that would be great. Okay, I couldn't wait to.
Thanks for a really interesting talk. I don't know anything about this field at all. I found the talk fascinating. But I did recently read the book called The Coming Wave by the DeepMind founder. And I don't know if you...
I haven't read it. No,
okay, so
I must.
In the book he says two things, and I've no idea, but I'd be very interested to hear your opinion on them. One, he says gain-of-function research is not worth the risk, just because there's been a lot of reported incidents in, is it H4 labs, or like whatever the top class of thing. He says there's just been a lot of reported incidents. I think he says like 70 in the last few years, and he says it's just a matter of time before someone screws up. I'd be interested in your opinion on that. And the second thing is, he says with the lowering costs of DNA synthesis and sequencing technology, he seems to think there's a material risk that the next pandemic could be started by a bad actor. And is that actually feasible, or do you think that's just like Hollywood and sensational pop science stuff?
Yeah. So, I mean, great points, and thank you for the challenge, if you like. So, yes, we work with these viruses in containment level 3 or even 4 labs. The incidents that they recount are over decades, and there has not been a release. There hasn't been, an onward situation. So, yes, everything is reported everything is carefully monitored and regulated, and that's why you know that there have been 70-odd incidents. But those incidents may be a spill which was contained but is reported. So if you do a sort of a public information search, you can find they're there for the public to know about. But I wouldn't say that that is, let's say, outweighing the potential benefits of working with such viruses and understanding the viruses, because without really understanding the mutations and the mechanisms behind them, I don't think we can improve the vaccines and be ready in the way that we could be. So, I mean, by all means, I agree that we should have international oversight, and I think we need to visit how we do that. But it is a dangerous situation. For example, one country, currently the United States, for example, shutting all that sort of research down, whilst other countries will continue to do it. And as you say, bad actors may also continue to do it, because I used to say it was very difficult to make a virus. I've been making viruses in the lab for decades. The technology came about in the 1970s when the first polio viruses were made just by bits of DNA into cells. It's not very difficult to do these days, as you say. Gene technology is there, and many people could do this. So better that we do it in a regulated way in the lab and really understand what we're dealing with, because otherwise there is a danger, yes, that somebody could make one. However, with all of that being said, nature is out there doing that experiment millions or billions of times every day. And there are many other things that we could look to regulate, like the way we farm animals, which are much greater risk and much more likely. Every single pandemic in the last 120 years has come from animals which we have had close contact with, farmed animals. So we should regulate all of these things. We should discuss them. We should regulate them on an international level and be aware, because this, viruses, as we see, go around the world. So it's no good one country doing one thing and one country doing another. We need open discourse. We need international regulation. But don't shut the scientists down at the risk of losing the knowledge that can be brought. That would be my answer.
Okay. Other questions? Melinda, yes, please.
Okay, I have lots, but I'm going to ask the one that might be more interesting to the audience. It was a brilliant talk. And I would say...
Speaking to the mic.
No,
it was a brilliant talk. And just as initially a comment, and I think the more that people like you talk to people like us, the more the public will trust scientists, because I think it's not just about regulation, it's about the public understanding that scientists are human too, and we have conscience. But the question, why is this virus so good at hiding symptoms from us? Why is it that we can get to the point where we're putting out maximum viral load, and yet somehow our immune system's not kicking in and going, I've got a screaming headache?
Mm-hmm. Yeah, I mean, in many ways, that's the perfect virus. I mean, viruses aren't designed to make us sick. They're designed to get in, replicate in our bodies, and get out again. And we are infected with many viruses which don't really make us sick at all, particularly when we're healthy. In fact, the most successful ones like the ones Colin works on, are particularly sort of silent, really, in us. So the immune pathology is when the immune system has got some sort of imbalance and it goes wrong. And so, the tests show, for example, that more than 14% of the severe COVID cases in the first wave were accounted for by people not making a very good initial immune response to the virus. So actually our immune systems work really well. The virus continues to replicate quickly, and I think it's about speed. I think the real issue is that many of the most successful viruses that cause these respiratory infections, at least, are in us and amplifying in us very rapidly and out again. And even when we do get sick with them, you may have seen with some of those participant scores that I was showing in the slide, the symptoms come late. The symptoms are not controlling the virus. They're just the bystander, the mess that's left behind of the destruction that the virus has caused in your nasal epithelium lining. And then you've got sort of, a cough and a cold and a tickly throat because your cilia are all stripped off and you're not capable of removing the dust. So in many ways, the symptoms are irrelevant to the virus. The virus doesn't need them at all. It's not part of evolution. There's no evolution driving the symptoms. And the best viruses are the ones which are in and out before you even know it.
Okay.
You mentioned that there were some individuals who are actively injected with the virus but didn't get infected. Could you tell us a bit more about that? Are those individuals being studied?
Oh, yes. I mean, the beautiful thing about these human challenge studies is that there are teams and teams of people, both here in the UK and all around the world, who've received the samples and are pulling them apart in every way to try and understand all of those different questions. So it looks as if some of those people at least may have in recent times been infected with a seasonal coronavirus. So you may know that the coronavirus family, there are these ones in animals which can jump in and cause these horrible pandemics, but there's also common cold coronaviruses, which are 4 different flavours that cause about 20% of all the colds that we get every winter. And it looks as if some people, probably in the winter of 2019, 2020, had one of those common cold viruses, and their immune system remembered some of the features of that virus which were similar enough to the SARS-CoV-2 virus that there was protection. It looks as if there was a tiny little blip of replication, and then it was completely squashed by the immune system. So those immune systems and immune correlates are being pulled apart at the moment, because if we can work that out, we may have a very good alternate route to vaccine, because the current vaccines, you probably know, make antibodies. All of our volunteers were antibody negative. They were totally naive to the virus, no vaccine, no prior infection. So something else in their immune system was helping them fight it off very quickly.
Fascinating talk. I have a question regarding the first part of your talk when you introduced the mutation. I don't know what the protein was called, ANP32. Yes. So you made it a single amino acid substitution, but the viruses are very adaptable.
Yes.
So is this a strategy that can really be successful to create animals that might not transmit or replicate?
No, I mean, you're quite right. So what we did in that first experiment was that we made the minimal mutation that the lab data told us might work. And we partly did that because the concept of gene-edited animals is, a little bit foreign to the public. And the idea was that, chickens themselves differ one from another by about that much anyway. So it's not like we were creating anything that was very different than nature would create. That sort of amount of variation we were putting into the animal was what you would expect to see in a flock of poultry anyway. It wasn't enough. It wasn't robust enough. That mutation we put in allowed a little trickle of virus replication, and then, as you quite rightly predict, the virus mutated and adapted to the broken protein that we'd made, and now is using the broken protein instead of the original one. What we think we can do is take the protein away altogether, and in fact we've already made chickens which don't have the protein at all, and they will be tested a bit later this year in challenge with virus. It may even get more complicated than that. In an ideal world, we might make a few different edits, because what we know, if I use the analogy of drugs against HIV, the cause of AIDS, when those drugs were first introduced and used in humans, a single drug was not enough because the virus evolved and escaped very quickly. But if you use 3 drugs at once, the virus has nowhere to go, so long as those 3 drugs target 3 different things. And so now people live with HIV because they take those combination drugs all the time. And in a similar way, I think with gene-edited animals, we would want to have several layers of protection, if you like, against the possibility that the virus could evolve.
Yeah, thanks for the great talk. It seems like a logistical nightmare organising this many people to work together, especially internationally. Do you think the UK response to COVID would have been better if we were still in the European Union?
Ooh, that's very interesting. I mean, I think we did pretty well. I think that the world's response could have been better if we'd have all talked a little bit better about the science as it was evolving. I had the honour to be at a sort of follow-up conference of science advisors at which many European colleagues were present. And to give you an example which is pertinent to what I've tried to say today, in the early days, in the summer of 2020, we were trying to advise how long should a person stay at home if they were infectious for. And was it 5 days? Was it 7 days? Was it 10 days? And we had very little evidence to know what the right number was. And most of the studies didn't come from our human challenge study because that hadn't happened yet. So it was from studies in the community, and everybody was different, and the route of exposure and the timing of exposure was so different. So we were blind. And of course, we put out one advice and the government put one policy forward. But if you went to France or the Netherlands or Denmark, their numbers were different. And the public then lose trust in scientists because they say, well, how come it's 7 days in England, but it's only 6 days in the Netherlands? What, you know, these scientists don't know what they're doing. So honestly, a coordinated, debated view of how to interpret the science and how to put out that message, I think is so important, because otherwise we're just going to lose the trust.
Thank you for your talk. You mentioned about the fact we're still having these waves of different strains. When do you think we could consider ourselves finally out of the wood, so to speak, with this pandemic? When will it just be like a seasonal cold?
Well, one could argue that it already is. I mean, so the sad thing is, I think, that we lost a grip of SARS-CoV-2. Firstly, early on, because we couldn't see who was infected. And then secondly, if I'm totally blunt, we did have, in the first wave, vaccines which not only protected people against severity and death, but also against infection and transmission. They were working very well when they were a perfect match to the virus that emerged. But we didn't keep up with that. We said, okay, we're only interested in protecting against severe death, severity and death, and that was enough. And so now the virus is disseminated around the world in such huge numbers. We will live with this virus. Humanity will live with this virus forever. It will never go. And what that means is that we can expect continual waves. I think we're all surprised by the frequency of each variant. I mean, normally with flu, we're used to one every 2 or 3 years, and at the moment with these variants, because there's so much evolutionary opportunity, we've got one every 2 or 3 months, it seems. Not all of those require vaccine update, but we are having to now update the vaccines and chase after the virus just the way that we do with flu every year, and it's a huge cost and unsatisfying way to deal with it. Thankfully, at the moment, the Omicron and all its lineages, which are all that's left, appears to be a milder virus. However, if you are elderly with a failing immune system, immunosuppressed, or taking certain medications, that virus is still very threatening to you. And there are, twice as many deaths now from COVID derivatives as there were even from seasonal flu each year. So we've tripled the burden on our hospitals as well by not getting under control. So when will we rest assured that a dangerous new variant isn't going to come? I think I have to say never. We will never know that. The virus is also seeded back out into wild animals, white-tailed deer, and who knows where else, and in many long-term immunosuppressed people with chronic infections. So new variants with quite different behaviour could emerge. Fingers crossed, it hasn't happened yet so far.
We'll take one last question. Colin?
So in terms of preparedness, I mean, it'd be fabulous to have lateral flows in everyone's bathroom cabinets which test for every possible pandemic that's coming. I mean, 2 questions. Obviously, how practical would that be? I mean, you probably focus on the respiratory infections like flu and corona, but is it even feasible to have something that will cover everything that could emerge from the animals? And also, there's always a problem that comes in terms of health economics. How much investment is put on this compared to everything else? Yeah,
I mean, so I think, of course, one would love to have a broadly acting antiviral. Thus far, they haven't really hit the success bar. But the antivirals that we have against COVID are effective against every strain of SARS-CoV-2 and actually all coronaviruses, if we're honest. The antivirals we have for flu are effective against all strains of flu. So I think that with a limited number of antivirals and diagnostic strips in your bathroom cabinet, you could be prepared for the most likely, so far, scenarios of respiratory pandemics. I haven't even mentioned, of course, where we're going with climate change and vector-borne disease. Do we need this for dengue as well? And where are we going with that? That's another whole story, which I'm not well qualified to comment on. But in terms of health economics, I mean, I guess I would just turn you back to, what was the cost of the COVID pandemic?
Okay, so in a minute we'll be moving over to the hall for drinks and dinner. But before that, I would like to thank Professor Barclay again for this fascinating talk. Thank you very much.